Hi, my name is Katharina Alamany. I'm doing my presentation on the blood-brain barrier, also known in this presentation as BBB. Blood vessels are critical to deliver oxygen and nutrients to all of the tissues and organs throughout the body. The blood vessels that vascularize the central nervous system, or CNS, possess unique properties. They are termed the blood-brain barrier, which allow these vessels to tightly regulate the movement of ions, molecules, and cells between the blood and the brain. This precise control of the CNS homeostasis allows for proper neuronal function and also protects the neural tissues from toxins and pathogens, and alterations of these barrier properties are an important component of pathology and the progress of different neurological diseases. The physiological barrier is coordinated by a series of physical, transport, and metabolic properties possessed by the endothelial cells, also known as the ECs, that form the wall of the blood vessels. And these properties are regulated by interactions with different vascular, immune, and neural cells. Understanding how these different cell populations interact to regulate the barrier properties is essential for understanding how the brain functions during health and disease. Some key information on advantages to the blood-brain barrier. The blood-brain barrier keeps the toxins and pathogens that are in the blood from entering the brain and spinal cord. While it prevents the toxic particles from entering, it still allows water, gases, glucose, and hormones that are needed for the brain and spinal cord to enter the system. This protection is an advantage as it keeps the brain healthy and helps to maintain homeostasis. Endothelial cells form tight junctions that act as tightly knit barriers, making it hard for molecules to get through. Although these junctions can be beneficial, if they are damaged, it can result in a bacterial infection. This is an advantage as it maintains a safe and constant environment in the brain. Throughout most of your body, a tiny blood vessel, capillaries, are lined by endothelial cells. But substances can seep in and out of them between the gaps in the cell layer. In the brain and spinal cord, the endothelial cells are tightly joined together, and substances can only cross the barrier through controlled transport channels or under special circumstances. The blood-brain barrier, BBB, is maintained by glial cells, including astrocytes. While the blood-brain barrier has great advantages to our systems, there is some key information on disadvantages. A major disadvantage of the blood-brain barrier is also its specificity. Throughout most of the body, passing in between cells allows molecules and substances to enter different areas, such as substances entering capillaries from the interstitial fluid and vice versa. This is a mechanism that many pharmaceuticals use to enter target locations. While these junctions allow better conditions for the brain, a significant disadvantage is that it greatly hinders the effectiveness of drugs used to treat various neurological disorders, such as Alzheimer's disease. The vast majority of potential drug treatments do not readily cross the barrier, posing a huge impediment to treating mental and neurological disorders. Despite the enormity of the societal and health burdens caused by Alzheimer's disease, there have been no FDA approvals for new pharmaceutics for this Alzheimer's disease since 2003. This profound lack of progress in treatment of AD, Alzheimer's disease, is due to two problems, both related to the blood-brain barrier. First, 98% of small molecule drugs do not cross the BBB, blood-brain barrier. And about 100% of biological drugs do not cross the blood-brain barrier. So, blood-brain barrier drug delivery technology is needed in Alzheimer's disease drug development. Second, the pharmaceutical industry has not developed blood-brain barrier drug delivery technology, which would enable industry to invent new therapeutics for Alzheimer's disease that actually penetrate into brain parenchyma from blood. In 2020, less than 1% of all Alzheimer drug development projects use a blood-brain barrier drug delivery technology. The pathogenesis of Alzheimer's disease involves chronic neuroinflammation, the progressive deposition of insoluble amyloid beta or tau aggregates, and neural degeneration. New drugs that both attack these multiple sites in Alzheimer's disease and that have been coupled with blood-brain barrier drug delivery technology can lead to new and effective treatments of this serious disorder. Researchers are looking at ways that they can utilize receptors within the BBB. One way is to create brain shuttles that modify antibodies to bind to these receptors, thus allowing the passage of larger molecules. If this method is researched further and clinical trials continue to be successful, this would be beneficial for future use of treatment drugs for neurological disorders. If the blood-brain barrier is damaged or weakened in some way, the immune cells are able to cross. These cells will then attack the myelin around the nerve, which then lead to near damage and can cause symptoms of multiple sclerosis as well, which is another type of neurological disorder. It is thought that there are a series of events that lead to multiple sclerosis. A breakdown in the blood-brain barrier is thought to be an early stage in this process. So now let's take a deeper dive into the multiple sclerosis side of things. If the blood-brain barrier is damaged or weakened in some way, immune cells are going to cross. These cells attack the myelin, like we said, and the nerves, which lead to nerve damage and MS symptoms. The blood-brain barrier can be damaged by multiple things. For example, stress, inflammation, or chemical processes are thought to trigger by disease, drugs, air pollution, or smoking. Possible links between gut health and the strength of the blood-brain barrier have been explored working on the microbiome. Several of the disease-modifying drugs currently available or in development act on the blood-brain barrier itself, or aim at stopping the immune cells from passing through the barrier. These include some drugs which bind to immune cells so they can't get through the barrier, others that target receptors on the blood-brain barrier to strengthen this barrier, and also trap immune cells in the lymph glands so they can't cross into the central nervous system. So in summary, some key takeaways that we should have from the blood-brain barrier information and its work on the central nervous system. The central nervous system is vascularized by the dense capillary networks that is critical to delivering the oxygen and nutrients we were talking about, and remove carbon dioxide and other waste products from the neural tissues, as from everywhere else in the body. These blood vessels contain a series of properties which we call the blood-brain barrier, which distinguishes them from vasculatures and other tissues and enables the central nervous system vessels to stringently regulate the transfer of ions, molecules, and cells between the blood and the tissue. This is critical to maintaining the brain's homeostasis, which allows for proper functions. So this is the main advantage, the wonderful part of this barrier. And it's very important that we have it. And it also works to protect the tissues from injury and disease and many neurological diseases associated with the blood-brain barrier dysfunctions. So it is important that it works well. This includes traumatic brain injuries, Alzheimer's disease that we talked about, stroke, epilepsy, and multiple sclerosis that we've spoken about. Therefore, a better understanding of the mechanisms controlling the development of the BBB may lead to improved comprehension of the pathophysiology of these diseases and further aid in the identification of targets to modulate the barrier to treat different neurological diseases. Many of the properties of the barrier are possessed by the endothelial cells that we talked about, that form the wall of those blood vessels that we talked about, but are acquired through a series of complex cellular interactions with the microenvironments throughout its development. These are some of the references that you've seen on the slides. Thank you for listening.